All right, everybody. Hot topic of the day is viruses, or as we like to call them, because they're keeping us stuck inside, stupid viruses. Anyway, that's our next lecture is viruses. I know you guys can't wait to get into this. One of the things I'd like to say before we go into this is that even though this thing can be quite scary, I think sometimes knowing a lot more about something actually calms you a bit. You know what can get you in trouble and what can't. I think the more educated we are about this, the better off we'll be. So we'll go forward with this. So I'm going to erase our title, our fancy title. Once again, you can see the setup. I've got the same notes you're seeing up here in the corner on the iPad. This is my writing area. I'll write my little diagrams and stuff like that, etymology of words, things like that. And we'll be good to go. Follow along with your notes that you have. So, basic info for viruses. A virus, and you can see there's a whole variety of shapes that viruses can come in. There's not just one. The coronavirus, actually, the very first picture looks kind of like what a coronavirus would look like. Something very similar to this, or this up here would be like a coronavirus, right? These things here, these spheres with the crowns or the protein extrusions on the outside here. Anyway, a virus is a non-cellular piece of genetic material, either DNA or RNA, and it's surrounded by protein. And that's all that's to it, is the nucleic acid, either DNA or RNA, and protein. There's no carbs, there's no lipids, there's not even any cells. It's non-cellular. They're not part of the six kingdoms, because if you remember, right, there's 5 features that you have to be a living thing. We went over that just before school had stopped for the March break, and I believe they show up again here in our PowerPoint, so I won't go into them right away. How do we categorize them then? Because we use physical features and stuff like that with the animals. We look at their cells, prokaryotic, eukaryotic. If you're not made of cells, you don't do a lot of the stuff that living things do. How do we categorize it? Well, what we use is their structure, their shape, and the type of host cell that they infect. And the word virus, etymology coming up again, means poisonous slime of plant or animal origin. So if you look at it, a lot of our viruses come from they mutate and they become transmissible. Means they can be passed on to humans from other animals. I believe the coronavirus that we're now dealing with came from a bat. So here's our slide to live or not to live. So they're currently not part of our 6 kingdoms. So I guess they're not living the same way we're living. Dogs are living, the plants in our yard are living, anything like that. But they obviously have a huge impact on living things. So, evidence that it's living. Well, it has biomolecules, like we said, proteins, nucleic acids, like DNA, RNA. They're there, and they're in us, and they play a huge role in us, as they do here. So, that's kind of a check mark to say, maybe it's living. It has DNA or RNA, and DNA is the blueprint of life. So far it looks like these things should be categorized as living. They are capable of directing their reproduction. Reproducing is one of the things all living things do. Viruses are masters of it. And they can evolve. This DNA or RNA can mutate randomly and create new forms of the viruses or change their abilities. And they're responsive. If conditions are good, they'll replicate faster. If conditions are bad, for example, and antibodies are released by the body, then they have a tougher time and perhaps die. Why they would be considered not living? Well, one, the cell, as you remember from much younger days, is the basic unit of life. They're not made of cells. So if the cell is a basic unit of life, well, too bad. They cannot grow or reproduce outside of a host cell, right? Other animals and plants and fungi and bacteria, they can reproduce on their own, right? Or with a partner, but you get what I'm saying there. So viruses, if we just had a virus and no other cells around it, the virus could not replicate. We could have a ton of viruses and they still could not replicate. They need the host cell to do that. And this is why we're isolating. We don't want to come into contact with other people with the virus where it gets into our cells. If we stay away, we don't get the virus. It's that simple. It can't run protein synthesis, and we remember proteins are the working molecules of the body. They run and regulate and build the body. Well, they don't make proteins. They use them, but they don't make them on their own. They use our cells to do that. So they kind of cheat. And they lack enzymes for cellular respiration. Cellular respiration is how you make your energy. These things don't run cell respiration like we do, like your cat or dog does, or anything like that. Now, there are 2 life cycles that these viruses undertake, and there are little nuances to each different type of virus. I'm actually going to go in. It's not on our lecture, but I'll talk a little bit about the coronavirus at the end of this lecture once we go through the PowerPoint notes here. So the lytic life cycle, before we go into that, lytic. If we see something lytic at the end of the word, at the end of a word, or lysis, it means to break up into pieces, to digest. And digest means to break up into pieces. When you digest your food, you're taking big chunks of food that you bit off of, a carrot or a hamburger or whatever it happens to be, and you're breaking it up. Digest means to break up into pieces. So if you think about it, that kind of makes sense. In grade 10, you had the thing, the lysosome. We now know that this is body, and this is something that's going to break stuff up. Excuse my lousy penmanship. But this is a little body, a little organelle inside of the cell that goes around to old and defective parts of the cell and uses hydrolytic enzymes to break things up. Oh, you're not working anymore? Chop, chop, chop. It gets broken up. Lysosome, right? A hydrolytic enzyme, which is found inside of the lysosome. Again, there's our breakup. And this means that the enzyme uses water to cut things up. So a lysosome uses water and its enzymes to destroy things in the cell that shouldn't be there, that aren't working anymore. So the lytic life cycle means something's getting broken up, and the bad news is what ends up getting broken up is our cell. At the conclusion of this lytic life cycle, the cell is ruptured and explodes, releasing tons of viral particles to go out and infect more cells. So let's take a look at this. Very simple steps here. So we'll start here, and then we'll work our way around clockwise on this diagram. But the first thing is attachment to the host cell. Now, some viruses are very target specific, like the AIDS virus. The AIDS virus comes into our body, and if it runs into a skin cell, nothing. Lung cell, liver cell, spleen cell, nope. It looks for T cells, a type of white blood cell. That's the only one that the AIDS virus wants to go into. So it's very highly target specific, right? Other viruses are broad spectrum targets, broad spectrum killers. They target everything. Ebola would be an example of this. The Ebola virus will go in. Hey, you want to infect the lungs? Yep. Kidneys? Sure. Liver? That too. Heart? Yep. It'll go after all different types of cells and tissues. And again, run a life cycle that results at the very end of that the cell explodes. It ruptures. So the first thing we're going to do is we're going to land on top of the cell. All right, so here's our virus here. Now this is a bacteriophage virus, right? If we look at it, here's our bacterial cell. I'm just drawing this corner part here. There's our DNA here. This is our bacterial cell. This is the DNA. And bacteriophages kind of look like lunar landing modules. They kind of look like this. They've got these tail fibers that come like this tube, and then they've got this part. This would be where the astronauts kind of, right? This thing that lands on the moon there. And what happens is inside of it, the virus, you've got viral DNA or RNA. We'll use DNA in this example, right? So here's our virus. And what's going to happen is this virus on the bottom of these little tail fibers has proteins. And these proteins match up with the outer surface proteins on a cell. So like I said, if this was an AIDS virus, even though it's attacking bacteria, the AIDS virus has proteins on its outer surface that match up with the proteins on the outer surface of the T cells, the white blood cells in our body. And it's like a key and a lock. And it says, hey, you can come in, right? Ebola, well, its key is like a master key. On the Ebola virus, and again, this is a bacterial cell, but the Ebola virus, the proteins on the outer surface match up with lots of our proteins, right? There are proteins that are on the outside of our cells that mark us as humans, and they're on lots of cells. And that's the key that the Ebola virus has. It matches that lock, and it gets in, gains entry to tons of cells, which is not a good thing. It kills us very, very quickly. So the virus recognizes, hey, this is the cell. In this case, a bacteriophage. This is a bacteriophage. Bacteriophage literally means, the bacterio is bacteria, obviously. Phage means to eat. Sorry for the lousy penmanship again. But this is an eater of bacteria. This virus is going to infect this bacterial cell and end up killing it in the process of its replication. So this outer area is our protein part, this capsule, and on the inside is our nucleic acid, in this case DNA. Right? It can be RNA as well. What's going to happen is, and you can see it here in the diagram right here, only the DNA goes inside of the cell. DNA is double-stranded. I'll try to draw that nice and neat. The DNA goes inside of the bacterial cell. The protein outside the carrying case doesn't go inside. Only the nucleic acid, in this case our DNA. So the DNA, the viral DNA, goes inside. And now inside of the cell are these little organelles that do things that the cell needs them to do. And what the viral DNA does is it says, hold on. Everyone stop. Don't listen to this DNA anymore. I'm the DNA in charge. So you're going to do what I say. You're going to build what I want you to build, right? So this DNA goes in and takes over this viral DNA. And we don't listen to the bacterial DNA anymore. And so what happens is, and you can see it in the diagram here, the viral DNA says, here's some new instructions. Start building this. And so what we build are a bunch of parts. Those parts are for more viruses, tons of viruses, hundreds of them. And then we assemble the parts. You can see that here in our PowerPoint. The bottom part, D, we assemble those parts. We put the viruses together. We just built the parts. We assemble them. The last instruction that the viral DNA gives, or RNA gives to the cell, is here. I got one last thing for you to do. Make this enzyme. It's all good. It's all good. Trust me, make this enzyme. And what that enzyme does is it eats a hole inside of the bacterial cell, in the cell wall. And what that does, well, think of the cells being like water balloons. They're spheres filled with fluid. If I take a pin and make a hole in that wall, that water balloon explodes. And that's basically what happens to our cell. That last little instruction to make that enzyme, it ruptures our cell wall. And now all these viruses can go out. And one virus that got in can make up to 100 viruses quite easily. And now 100 other cells could be infected. So this is how viruses replicate. So if we go to our PowerPoint note here, we look. We have the attachment here, the recognition between the viral proteins on the outer surface and the cell outer surface proteins. The viral DNA, I use DNA here, could be RNA as well, but DNA is injected. Takes over, tells the host cell's machinery, hey, don't listen to your own DNA, listen to me. Foolishly it does. We build up a bunch of viral parts. Those parts are then assembled, and then the enzyme is created that destroys the cell wall, and this is the lysis. Right? This is why it's called the lytic life cycle. Lytic or lysis means, like we said before, to break up. The cell breaks open and all those viral copies are released into the surrounding environment if we're talking bacteria, or our bloodstream or tissues if we're talking inside of our bodies like the coronavirus. There is another life cycle. The second one, I'm just going to erase my diagram here. And so the second one is called lysogenic. So there's lyso again. I know that means to break up into pieces, and that's what happens at the end. But then we've got genic here. And we've dealt with genic before. We just did genetics. And genic means generation. One after the next, after the next, after the next. So that's what this is coming up with. That's what we come up with for this. Generations. All right? Parents, kids, their kids, their kids. So we're going to be seeing that here. And then, of course, life cycle means how this thing lives and continues to produce the next generation. So think of this. The last one was just break up. Right? Virus recognizes a cell, throws in its DNA, takes over, makes the parts, assembles the parts, boom, rupture the cell. Here's the difference here. There's going to be something different here that deals with generation. So thinking ahead, I'm going to be looking for that. So again, I'm over here now looking at this. The virus attaches to the host cell. Well, that's the same as before. Viral DNA or RNA, depending on the type of virus, it's injected. That's the same, nothing different. Here's the difference. This is the generation part of the genic part of the word. The viral DNA doesn't take over right away. And the other life cycle, the viral DNA couldn't wait. Gets in there, hey, stop what you're doing. You're listening to me now. This type of virus, the DNA actually becomes part of the bacterial chromosome. So if we were to kind of draw that, here's my bacterial cell. There's my bacterial DNA. So this is my bacterial cell. This is my bacterial DNA. The virus comes along. There's our virus. I'll draw its DNA in green, so there's a good contrast here. So there's the viral DNA, and it comes inside of here and doesn't take over right away. Instead, it embeds itself in here and just sits there. It lies dormant. Doesn't take over, just sitting there, just chilling, right? So now it's part of this circular DNA, the bacterial chromosome. And what happens is the bacteria just goes along being a bacteria, doing bacteria stuff, but part of being a bacteria is running binary fission, that form of mitosis where it makes more cells. So what happens is our one cell that is infected, sorry, the bacteria is orange, I'll use orange. The one cell carrying the virus divides and forms two. These divide and we've got 4, and then these divide. And you can see the numbers would go up really quickly. So through several generations, right, we can see here, we get a whole bunch of cells, right? Our one cell very quickly through a couple of rounds of binary fission or replication has made a whole bunch of cells here. Now part of binary fission is duplicating your DNA. DNA replication takes place. And so this was carrying the viral DNA inside of its chromosome. When that DNA gets copied, so it can go into 2 cells, this gets copied as well. So now the viral DNA is in here, in here. Doesn't say a word, just lies dormant, sleeping inside. Lets these cells divide. Now we got 4 cells with that viral DNA embedded in the bacterial DNA chromosome. Then it divides again, so it'll let the bacteria replicate several times, which produces a whole bunch of bacterial cells, right? Each of these new bacterial cells has now got the viral DNA inside of it. Then what happens is, the bad part, the virus wakes up, right? And now the DNA comes outside and says, now we're taking over. And now it switches back to that previous life cycle. It says, okay, we've done enough sleeping. We've been dormant long enough. Now we've got a whole bunch of cells, not just one virus-making factor. We've got a whole bunch of them. And now they all wake up. They all start producing viral parts. The viral parts are then assembled, and that enzyme is created. Right? The last step is make the enzyme that's going to destroy the cell wall and rupture these things, releasing the copies of the virus that were made. Now, instead of one cell releasing 100 viruses, we've got 8 cells all releasing 100 viruses. So 800 viruses or more, right? I'm using 100. It's not like we get to 100, it automatically stops. just using it as a number. But now 800 or more viruses are being released at once. So the virus gets inside of you and it's like, no, everything's good, everything's good, everything's good. And then suddenly it's not good. Now, if we look at this, steps 1 and 2 and 4, 5, and 6 are just like the last viral life cycle, the lytic life cycle. The only difference is this step, the embedding and sleeping. So step 3 is the only difference, where it gets in here and doesn't do anything for a while, lets the cell divide. So if we see a picture of that, actually this shows both, but here it is here. If we go along the bottom of this diagram, and this is why I put it in here, because it shows both of them at once. If we go along the bottom, here's a recognition and attachment. The viral DNA is put inside. And then, of course, it makes the parts, assembles a part, and ruptures. That's the lytic life cycle. This top part here, this extra loop, that is the lysogenic. So again, virus gets into the body somehow, recognizes the DNA or RNA, the nucleic acid goes in, and you can see here now, if we're going lysogenic, here, that DNA has become part of the bacterial chromosome. Then, of course, what happens is the DNA doubles, the virus replicates a bunch of times through binary fission, and we get a bunch of bunch of bacterial cells. Right? The bacteria reproduces a bunch. We get a whole bunch of bacterial cells with it, and then the viral DNA jumps out, says, I'm taking over now. And it goes back to what we talked about previously. Make the parts, assemble the parts, rupture the cell. So that's normally the end of the talk, right? I put there, right? When a virus thinks it's Thanos and wants to kill half the world's population, right? And toilet paper hoarders, they also annoy me. Don't be hoarding stuff. Don't be hoarding toilet paper. Anyway, quick little Corona talk. A couple of things first. They're talking about people being in quarantine if you went away, right? Quarantine. Actually, this is not a Latin root. This is an Italian word, right? Quarantina is 40 days. And what would happen is ships would be back in this is, I think, from the 1600s, so the 17th century, 1600s. Ships were the main way that we traded and sold goods, right? There weren't engines yet, so there were no planes, there were no cars or trains or any of that. So it was all ships. Or you had to walk and, with horses and stuff like that. So ships would come into port. Now, when they would come into port, they would be met by, the port master, the harbor master. And if they noticed that there was sickness on that boat, or it was known that, hey, that country, they're dealing with an epidemic right now, and it's isolated in that country, but this boat's from there. What they would do is they'd make that boat and the guys that are on that boat stay on it for 40 days to make sure that whatever sickness was going through it, if there was sickness, stayed on the boat. After the 40 days, the disease would have run its course, and then you could come on to land. You can imagine the boats then weren't exactly ocean liners or those Disney cruise boats. They were pretty crude, so that would be pretty tough. You'd have to be pretty tough to be a sailor. An interesting thing, if we link that back to the Bible, there's another famous boat that while the earth was cleansed, certain animals in a family, right? There was a guy that built this huge boat with all the animals, but I don't know his name right at the moment. Ha ha ha, bad joke, I know. Anyway, Noah's Ark, 40 days. The earth was cleansed. Kind of a neat sort of connection to that. I don't know if it's a legit one or whatever, but I kind of noticed that when I was researching some of this stuff. Anyway, so that's what quarantine means. It means you're isolated for 40 days. Now, we're just asking for 2 weeks now with the coronavirus, and some people still can't do it. Speaking of corona, there's another word. Corona means crown. That's a very terrible N. I'm going to redo that. It means crown. All right? If we look at this virus, right? It's actually a retrovirus, so it has RNA inside of it. So there's my nucleic acid. On the outside are these little things like this. These are protein, right? Remember, the outside is the protein carrying case. So it looks like this. Right? And so That's what a coronavirus looks like. And these are the proteins that it uses to recognize certain cells in our body, specifically the lungs. It's going right after people's respiratory systems. This matches up with a protein on the exterior of our lung cells, and that's what allows it to gain entrance. Now, what people want to do, or what our body normally does against a virus, or tries to do, is to create antibodies. Antibodies are Y-shaped molecules that go after things like viruses. So we have very specific cells, actually T cells in our body. And what the T cell does is it takes a picture. Think of a T cell as having its phone and going, oh, that shouldn't be here. This doesn't have my protein on the outside. And it goes like this. Pretend this is a cell phone. Click, and it takes a picture of this. It then runs to another type of white blood cell called a B cell. A B cell makes these antibodies, and they're Y-shaped molecules. They look like this. Now, all antibodies have this Y shape. The end of the Y, the fork in the road here, that's what's different between different types of antibodies. And these antibodies match up with this shape. That's why the picture was taken by the T cell. It'll run to the B cell and say, hey, make these things. I got a picture that matches it. And what the antibodies do is they go and they attach to the virus. I'm just going to draw a few antibodies here. So what they do is they attach to the virus. Now, viruses are very small, right? We've drawn this in class. If this is one of our eukaryotic or a human cell, like one of the cells in our body, a bacterial cell would be like this, right? So this is a human cell. This is a bacterial cell. A virus is like that. So this is obviously not drawn to scale, but there's our virus. It's very small, very, very small. So sometimes white blood cells miss it. They don't see it right away. When a bunch of these things stick to this, it does several things. One, it immobilizes these things. It kind of slows it down, right? It's like throwing a bunch of glue and concrete on this thing. It's going to slow it down and not allow it to gain access to the cell. And you can see it actually coats the outside of the virus. And so now that exchange between these proteins and your cell ones doesn't take place. It can't gain entrance. What it also does is it makes these things a lot more noticeable to white blood cells. And white blood cells, like macrophages and those things, can now go over and go, wait a second, look at this thing. It's got all these antibodies on the outside. That's not right. I know antibodies attack things that shouldn't be here. So when it sees these things all stuck to the outside of this thing, which is already slowed down and can't get in the cells, it allows the white blood cells to come over and destroy it. Right now, what's happening to patients is the coronavirus is getting inside of them. It's going after the lungs and their bodies. It's basically a battle of time, right? The lungs are the wimpiest organs of the body. When they get infected, man, it can really hurt you, right? Bring oxygen to the body. Oxygen is needed for energy. So this is why it's so bad. So what you have to do is you have to form antibodies, but it takes time. And in that time, if the virus gets the upper hand on you, you could die, right? In that time, you're very sick. But once the antibodies are made, then you can come back and you see people that are recovering. In fact, a lot more people are recovering than that are dying because their cells are making antibodies that are recognizing the virus now and helping their own body to defeat it. It's a form of active immunity, which means your body takes action. Now, another form of immunity is a vaccine. What a vaccine is a weakened or dead form of the virus. So if here's our normal coronavirus, right? It's sitting here going, shh, right? I'm healthy, I'm good and all of that. Well, what we do is once we map it out, we kill it. And we take a form of this virus that is dead or weakened. And we inject that into the body. So this is what we do more often, but dead or weakened form of the virus, right, or part of the virus, and it's put inside of the body. right? And what happens is our body reacts like it's the real thing. It's like, oh my God, it shouldn't be here. Take the picture, make the antibodies, immobilize a response. Quick, 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 do what you can. The good thing about a vaccine is because the viral particles are dead or weakened, it's not going to make you sick. So it goes in there and it just kind of acts as practice. But what we do is our body will have the same response against a much weaker, much less virulent intruder, and it makes the antibodies. And so when the real thing comes along later on down the line, you've got your antibodies already made, and you kill it right away. Right? Your body has memory B cells, and it's like a library or a USB stick, right? It remembers all of your previous encounters, and it keeps on file all of the shapes that it has to recognize and stick onto antibodies to battle these things. So that's why you only get chickenpox only once. You can only get measles once. You can only get certain things once. Once you've had it, or once you've had the vaccination, you can't get it again, because we have a library, a memory to our immune system. And so even if you did come into contact with it, your body goes, oh, we've dealt with that guy before. Antibodies are made almost right away, and you never really ever get sick. All right? Anyway, that last little bit is not something that was in our lecture. I know, but I think it's something that you can definitely appreciate going forward. It's nice to know a little bit more about what we're dealing with. If you go onto YouTube or do some research online, go to good websites. We talk about how to recognize a good website versus a bad one. And you can learn more about this stuff. But I just thought I would put that out there, and that's it. Anyway, hope you enjoyed the lecture, and I'll see you in the next one. All right, bye-bye.